And I was talking about a very specific experiment with case data, unrelated apparently to your data. And then the second part, I'm going to tell you about the experiments that Johan did. So this is a kind of slide that we saw this morning a little bit from Bill, but this is taken from a paper by Nancy Bleckner about 3 or 4 years ago. And the fact that the Y chromosome is so organized in some kind of legal fashion, it's not a very stiff helix and very stable helix, has been known for a long time, possibly even to Fleming, I don't know. And the impetus to get after this now is through these experiments on high-speed and more also directly imaging methods, which are in fact needed to fully sort out the structural details, including things like handedness, which will play some role in what I'm going to talk about. Loop extrusion idea was proposed, to the best of my knowledge, at least been most popular by this guy Tim Naismith. I don't know him either. But there are hints and papers on loop extrusions, even in the 80s, it turns out, and including things like using cross-linking to measure proximity between both sides. These are known in 1980. So what is loop extrusion? So the loop extrusion that I'm going to talk about is somewhat different, mechanistically speaking, and I'll tell you why. The loop extrusion that is shown here is due to Erez Lieberman and also Moni, more or less around the same time. And the idea is that these motors, and in the first several papers they were not quite sure what those motors were, they called LEFs or something like that. They would land on this chromatin thread and they would translocate while being attached to the chromatin, and then when they encounter some stop sign, which are some architectural proteins, the loop extrusion would stop, and of course the motors don't sit on the chromatin forever. There's a lifetime associated with that, and then the process can continue. So what I'm going to tell you about is a different picture, and it's called this crunching mechanism. I don't know if this works. Yeah. And this crunching mechanism, this is a cartoon that you should not believe at all, but Case Decker provided this to me, but you can download this. And the point is, here the motors do not translocate. They're essentially stationary. It's kind of seedless, and they're consuming ATP and things like that, as I'm showing here. And the time it extrudes the loop, and the loop essentially stops by some kind of roadblock mechanism. Okay? So the motor is stationary. So what then drives this loop out is an allosteric transition that takes place in the motor by movement of the engine. This is a motor, which means it has a catalytic cycle, consumes energy, undergoes conformation change, hydrolyzes ATP, and then it rejects the inorganic phosphate, and the ATP cycle sets itself again, and the conformation changes that take place for the amount of this. So the first steps, for example, could be ATP binding. We think, in analogy to motors that we know elsewhere, like anisine and myosin, that step of ATP binding is the power stroke. Whatever that means in this context, it's not been sorted out structurally extended. And then in that power stroke, the red guys, which are the hinges associated with the SMC complex, they make a transition closer to the head, and the head is, of course, roughly speaking, stationary in the process. Right? And it's a transition between the head and hinge that involves hydrolyzing ATP, maybe 1 or a half, 2, that in fact causes the allosteric transition, and that results in loop being extruded. This is an experiment that we saw this morning from Bill. This is case-based experiment where he patterns DNA at 2 points, and there's a flow. The flow generates a kind of force and supplies ATP, and then you see the loops extruding this big red thing in the middle, and you can decrease the flow rate at a fixed ATP, and then measure what the velocity of the extrusion velocity is, and as a function of force, and that's the curve that we're going to chase. And this is condensed, by the way. I don't remember 1 or 2, and I don't think any that there was 1 or 2, but the velocity is about 1500 degrees Celsius. So we want to understand this theoretically, right? The mechanism that we're using is because of the dynamic of this experiment is reported 20 years ago on bacterial ribosome transcription, where the polymerase is opening a bubble for the transcript to be synthesized. These are 2 back-to-back papers in Science. Richard Ebright is the author of both of them, and there are some very clever single molecule places. And so I want to read to you what it says. We show that the initial transcription proceeds through a crunching mechanism, in which the polymerase remains fixed on the promoter, in this case of DNA, and pulls on DNA onto itself past the center. So it's similar to the cartoon that I showed you. It just pulls it, and then the bubble opens in this case, and the loop is extruded. In fact, this paper made such a great impression on me. I walked up to my student when I was in Maryland, and in fact, we wrote a paper explaining this using computer simulations. So this mechanism was stuck in the back of my head, and when I saw Case's experiment, I thought this could be the way to think about this. Some little thing about condensing sensing motors. They all look the same to me, and the structures of not all of them are really known. So it consists of 2 heads, those are the ATP binding sites, and a hinge, and the binding triggers increase in the distance between the head and the hinge. The resting state, if you want, is 250 nanometers. Okay, we don't know much less about how these motors work, but we can explain and we just spend time. So how do I translate this idea of scrunching into a little bit of arithmetic? And the way it works is that we want to consider the DNA as the thread. The motors are here in this red and blue. I'm not so sure this is going to work. Red and blue. And when the head comes closer, the distance between the head and the hinge decreases. So the distance R2 is less than the distance you started. When that happens, it's triggered by ATP. It means that I have to bookkeeping, and I'll do the bookkeeping in a second. Then right? And so the loop increases some number, which is initially L1 or something to something else. So that's the process. So that physics, this physical picture, you have to translate it into some mathematics, and then it involves knowing a little bit of quantum theory. We didn't do any simulations here, just theoretical work, and I'm not going to bore you with that. So that's what is happening in the motor upon binding ATP, and I'm going to go with this, etc. So what are the energy costs? So I go to thermodynamics, and energy is of course conserved. And so in order for the loop to be extruded, if we impose some load on it, the motor has to do some work on that chromatin, and so that's what's called WF. Even in the absence of the load, because on the scale of 50 nanometers, so DNA is stiff, I'll have to do some work on it. So I'll have a total work that I do that the motor is whatever the external load you're imposing, that's the kind of experiment case taken with. And where do I get that energy? Because my ATP hydrolysis. So number, so I have the total number, and of course, the motor does not have unit efficiency. It's kind of like all our cars, and as a result of dissipation. So when I balance this stuff out, I've taken down care of the thermodynamics, the energy cost, I've taken care of the fact that there's a catalytic cycle, and I'm ready to, in fact, derive an expression, and it's easy to do. And then forget about this, the velocity, exclusion velocity is simply some factor times some distance. In fact, if you, in freshman physics, high school physics, you know this has got a dimension, F is the external force, that ranges from some number to some number you can see the second. So this is a theoretical expression, very simple, and it has two parameters, it's K0, that's it, I always say, essentially, and WR, that's a mean distance that decreases as the allosteric transition in the motor actually takes place. Okay, so how does this theory work? I have two parameters, I don't know what they are. Actually, I only need one, it turns out. On the left side, you see it's calculated velocity, a function of the distance, and the red dots are for cases of paper in science, some academic company, to KET, find out the data for the distance. So, and then you can read off what this K0 is, and it turns out that our predicted values are 5 times, or maybe 10 times larger than the experimental electrolysis rate, which I think has not been really measured very clearly yet. The other thing which is important is that each time you do this experiment, the extent of the extrusion length varies. So there's a distribution of these lengths, and that distribution is extremely broad. It was first reported by Arano, and also there's a report in Case Deckers. So the theory, no parameters anymore, except for the ones that I showed you, reproduces the distribution extremely well. What is remarkable is, even though the motor maximum distance is about 50 nanometers, it can extrude loops up to 150 or even 200 nanometers. And that's really a little odd to understand from the structural point of view. I'm going to skip this. I'm going to skip this. We can say a lot more about this on the data. So now I'm going to come to my thesis, and this is an experiment that probably Johan might show, and AW showed this to you. And this is this paper, well known, apparently 7 years later, there's another more well known paper. Works or it's not by our right? And so I'm going to skip this because we recorded this, and my thesis is all this different process. But here, the motors cannot consider just a single motor. They need to consider multiple motors. And these are numbers for the condensing 1 and condensing 2 that you get from Gila cell. And you can see that the amount of time is bound. Condensing 1 is bound to the drop rate is 2 minutes, whereas condensing 2, the residence time is about 2 to 3 minutes longer. So you naively would think, and correctly so, that the loop lengths that are extruded by 2 is in fact greater, and this is something that we calculate. I'm going to skip this. Alright. So this is things that maybe Johan will tell us more, but this is the high-C experiments. But we love the paper for the following reason. They did experiments with and without condensing 1. They did the experiment with condensing 1 and condensing 2 and together. Okay? And what they find as you do this as a function of timeless contact probability, we see that when you have both of them present, which is the wild type on the left-hand side, there's a little bump that we also showed this morning from Bill's sentence that's much more pronounced than that, and that bump keeps shifting to the right, but it may plot this distance in distance. And when you do this only with condensing 1, for some reason it's called cat-based, I don't know, and then you don't see the bump, and therefore they're rightfully attributed that most likely the helicity or something like this is coming. Okay. Bill, I only have 5 minutes here. Huh? I'm going to skip this because we developed how to think about how multiple motors could in fact act on this is a generalization of the single motor that I showed you before. But from this, you can calculate this PFS, or contact population functional genome distance.
Here, 9 minutes.
Oh, 9 minutes.
No, 4 minutes. 9 minutes total.
Okay, so in that case, I'll show you this very fast. This is how multiple condenses work. When two condenses in fact meet, they pause, and there's a pausing time, which is the parameter in the theory, but typically about 7 seconds, because somewhere we found that's somewhat a number, you can change it very easily. So this is a single realization of multiple motors. So the loops are extruded, they're stable to the corners, and if you look at the axis at the end of this one such experiment, you have a roughly axis, but that axis is very dynamic, because next time you do that experiment or simulation, it's going to be different. So the axis is not a very rigid helix at all. Okay? So you can once again calculate this analytically. Okay, so when you do this multiple motors, you calculate the contact property as a function of genome distance, and this is sort of wildfire, like a couple of cells and newer cells, blah blah blah, and the calculations in fact are pretty good compared to this practice. So things the big but then there's this nagging experiment that Johan did, when he was only condensing one or only condensing two. And these are the kinds of structures we generate in this active motor with multiple guys. You can see helices and stuff like that. So we have a pretty good agreement for the only condensing one with this multiple motors, but when we try to do this with condensing two, our theory is not very good. In particular, the all-important bumps that he's getting at a big kilo, we cannot do this. We're still thinking about why not, so we did something else. So I'm going to skip this. We developed a theory some years ago, 3 years ago, where we could take three high-C maps, any high-C map at any resolution you give us, and we use polymer theory, again there was no simulations there either, and then turn that into three-dimensional structures for each locus. So there are 3D structures, it's no longer a contact map. Okay? So in the 3D structures I can calculate anything I want, and that's what I'm going to show you. So that method is called HIPS, or the way it's called HIPS, is high-C polymer physics structures. And structures in the very much the sense that molecular biologists would use, except the resolution here is not as good. Right? So these are various PFS curves. Right hand side is the kilo cell. The others are no condensing 1, we condensing 2, we condensing 1, divided 5. Now you see that I can reproduce this quantitatively. I must tell you, this doesn't mean anything to experimentalists. There are no parameters in this theory. It's not technically simple, but no simulations for sure. Right? So this works pretty well. So now that we have the 3D structures, I can actually tell you, at least according to the theory as we have developed, which uses only as input the high-C contact map, right? People in this room can measure, some people can measure. I can tell you what is the nature of this helix, what is the handedness of this helix, etc. This is an image from Nancy Clayton's work. It's an ensemble library, and then she makes an argument that this is not really strictly a helix, but it's a helix whose handedness changes as you go along the axis of the helix. Remember, the axis is very dynamic, so the statement may or may not be correct. And this notion that the handedness changes, it's called perversion. This is known to Maxwell 100 and something, whatever my cellular, and it comes to mathematics. So, and in fact, you've seen this very often in plants called tendril perversion, and it's the same idea. And because we know the 3D structures, we can actually calculate very precisely what the angular correlation is. And that's what is being plotted here. There's some sort of function at the bottom. Tells you to sit here, march across the genome, go in some distance, and then see how correlated they are in terms of orientation. Right? And that's being plotted on the right-hand side using our method, and 30 minutes, which is what we chose. We've done it for other times as well. And I think the red guy, the wild type, and you'll see that the wild type is not that dissimilar from the condensing tube, which is the red guy, which is the blue red, whatever, right? And you can see there are periodicities, and the periods are exactly around 1 to 4 megabases, right? And if you look at condensing 1 on the other hand, which is the yellow extent, there's a minor peak, less than 1 megabase, and so it does not sustain the peaks, and that seems to be consistent with what people think it should do. So it's very clear that in fact there is this angular correlation, but it still doesn't tell you what the happiness is, because if I switch these angles, I'll get the same result. So I need to figure out what the happiness is. Because we have the 3D structures, and we have an ensemble of them, we can create 100,000 structures if we would, or more. We can look at each structure and define a local frame for the helicity. And in the way we define the local frame, if the parameter is 1 is right-hand helix, the parameter is -1 is left-hand helix. Okay? But if you see a conformation, the continuous 2 conformation at 30 minutes for 100 such structures, you see there's a huge distribution of this happiness. And I can look at them and say that the happiness, in fact, is changing, in fact, more randomly than you think it should. And the mean value of the happiness is not that different from 0 to the average. Right? For any given realization, I mean, a given structure, the happiness can be changed, and it's never 1 or -1 at all. You see the probability of it being at -1 is very, very small. So what does this tell you? That this is what we call random release correlation. This is a correlation because your happiness indeed changes so randomly that you can only predict if you do the structure or do the imaging. And this is the stuff from Maxwell, and none of the young people know this, but if you know the Lord, they like all this stuff. I'm done. I want to tell you about Darwin. Apparently, at some point, Darwin was sick. Somebody of his in the US told him about tendril perversion. And in his room, he actually grew this plants that, in fact, have this perversion. It's kind of shown at the top, it's kind of like a telephone cover. And then he gave a lecture that was turned into a book. That book has 0 illustrations, very inexpensive by the way, it's $5 or something like that. No illustration, nothing, and he just tells you about this experiments that he did with his plants. And this is what he writes about the importance of perversion in the plant system, and maybe I can read this. It says, I have more than once found on purpose to be gave the voice that growing in an exposed hedge. The tendrils attached to the surrounding bushes, kind of like that maybe. And as the thick and thin branches were tossed to and fro by the wind, the tendrils, had they not been excessively elastic, would instantly have been torn off the plant thrown across the way. But as it was, Pironi safely rode out the gate and the ship with 2 anchors down. Long range of cable I had to serve as a purpose as a spring actually surface in the storm. First of all, none of us I don't like this. maybe Bill, having lived in England, writes this book. This is really very beautiful and summarizes exactly what it's saying. There are some philosophical questions that I don't know about. If you think about symmetry being extremely important in nature, nature almost always prefers symmetrical stuff, then the random helix provision is not that symmetric, right? A randomly changed. It's a random structure, which is pretty unusual. So maybe it should be like a picture that Nancy Clinton or somebody has that is a regular helix provision. We don't know. We don't know that. We need really good imaging experiments to sort this out. And I don't know why nature would have, because this random helix perversion itself is an instability that nature should not want to tolerate, but maybe you don't want too stiff chromosomes. You want something that's flexible enough. And this is my singularly the greatest achievement as a scientist. This picture on the left-hand side is in the cover. It was made by my group member, and as a guy who is the group that deals with physics, this I think was a crowning achievement for us. That some reports in fact published this after 2 of the 3 reviewers said this paper, no one's some reports would read this paper at all. The editor decided to put this on the cover, and we're very happy. I'll be happy to take questions.
Do we have any questions for Dave? Okay.
Yeah, so I guess the thing that I didn't understand from the models of the second half of the talk is that you can generate these scalable structures, but what is the underlying physical mechanism?
So that's what the motor model was supposed to have done for us. It did for telecondensate 1, that it's extruding loops until it encounters something and pauses and continues. All right? But in the second, in the calculation of the structures, we just have a theory that converts high C to medium structures.
But how does bypassing lead to?
How about it work?
So if I understood correctly, you're saying that it's condensing, bypassing leads to the helix.
No. The second case I don't have in the second part where I showed you the structures, determine the structures from the high C. There is no mechanism that we know, although we know that condensate 1 is less important than condensate 2. So the reason we're interested in trying to figure out the mechanism was to explicitly include the condensate 1, hoping that we would get all the results from that mechanism, and that we did not get from condensate 2. Right? We still quantitatively understand these experiments using the algorithm that tells you what the precise is. And we're still trying to figure out why is it in 2 cases we cannot work it out, but in 1 case it's really a motor, multiple motors. You can't do this with single.
I have a second question.
Okay, go ahead.
So in the first part of the talk, we talked about the stretching mechanism.
Yeah, something, yes.
So how important, so I think that maybe I missed some details that they were omitted, but how important is it that it's scrunching as opposed to doing something like loop capture, like others have proposed? Or zipping or whatever.
Here's the deal, right? Using the scrunching mechanism in theory, we could quantitatively reproduce experiments, which is 0 doubts about that. I showed you. The onus is on other people who propose other mechanisms. I talked to your man Ronnie as well, to actually calculate exclusion velocities and distribution using any other mechanism that you can think of. I haven't seen it.
It doesn't have to scratch.
That's what we think happens, and it's sufficient to produce a theory that actually produces that. Right? It's possible there are other things, but somebody who proposes those other things, right, must do what we already did in order for me to take it seriously.
Okay, Dave, thank you very much. I think it's time for us to move on. Okay, we're now going to step a little bit away from the rarefied air of scary equations and move to the air of scary large-scale DNA sequencing and analysis. Johan Gitwitz from the University of Massachusetts in Worcester will tell us about some rules of engagement.
Oh yeah, thank you, Bill, for everybody hear me. Thanks to the organizers for having me here, and I must say I feel tremendous pressure now for the amount of times that my name is being called out here, and I hope I don't disappoint you with the story I'm going to present today. It's going to go into some of the things that have been mentioned before, and it is a wrap up our latest work that built off, as we like to call it, the rules of engagement for chromosome folding. Fleming's been mentioned a couple of times before, but what's remarkable about Fleming's microscopy work is that he already recognized in the 1800s that there are basically two different ways to fold the genome. There's an interphase and there's a metaphase. And since then, we have made quite little progress if you think about the amount of time that has passed. But right now, I think we're trying to get a better and better understanding of what's really happening. So what we are trying to figure out is how chromosomes are folded and which molecular machines fold these chromosomes and how they do it. The way you do this in the deck by using chromosomal information capture was already explained by Ed, but I'm going to step over this explanation to show you again the map that comes out of this technique where you basically plot the chromosome against itself and you can figure so this is the chromosome folding map for the interphase. This is what it represents, an interphase that moves. And as mentioned before, there are compartments that you can see on the map that represent themselves as heterochromatin and euchromatin in the interphase nucleus. We can observe other structures that are more detailed, like tabs, these triangular structures, also worked well by us. And within those tabs, we can even find actually not the case. These machines that are doing the work are all very similar. The cohesins and the condensins that have already been mentioned before. In interphase, we have the active cohesin, and in mitosis, we have condensin 1 and condensin 2. And the motors all work in a similar fashion, and with no restriction. They've also mentioned before, these are very familiar. I'd like to show you now a wet lab experiment by Johan Gitwitz, where he tries to explain what loop extrusion actually is, and he's explaining how with a double-sided loop extrusion, you can basically create a situation that we see as dots in the heat. And thinking about this, I thought this morning while I was looking out the window and strapping on my running shoes, because my wet lab experiment, I like to learn how to do it open, is that if you fold these kinds of loops, you can also do a single-sided loop extrusion that leads to the lines and flames that you can see on the map as well. So they're very similar loops. They're generated slightly different, and they give slightly different features in the 3D genome. So basically, these cohesins and condensins are all loop makers, or loop extrusion factors. And in the Murdy lab, people have Jeff Hugenberg Anton Brodko, John Hugenberg, and now people working in the lab are very good at modeling what the loop extrusion process. And if they do so, they basically recapitulate the high C maps that we find in our experiment. Now, I talked about the interface folding. Metaphase looks very different. The interface is this decline of contacts over distance, as you will see in this interface map. And these distances are actually declining rather gradually. Whereas the interface in the bottom left, or the metaphase, sorry, in the bottom left, there's a limited distance of interactions. And a very sharp decline at around 10 megahertz. And so that's a very, very different way of folding the genome. The question is, how is that possible if it's the same similar type of motors of machines that work on this chromosome? So in 2018, we found a couple of things that work. We found that to create a metaphase chromosome, you would require a helical path. And then within the axis of this helical path, there are two types of condensing. The condensers two are at the center, and there are nested loops, loops inside loops, generated by condensing. And so we have these pictures of interface and metaphase. But what I am personally really interested in my other work on differentiation, I kind of do the same thing. I'm interested in looking at the dynamics. So how can we see this process happen? So what we're doing is what people used to do in the 80s, when you take a photo camera and you take multiple pictures after each other, you can kind of create a video. That's what we're aiming for here. We're taking a lot of snapshots to capture the dynamics better than by taking a few photos. So onward from photo. Now first I want to stress that what we're going to do is we're going to look at the transition from interface to mitosis. And so we are going to end up at some point in prophase. And in prophase, there's a situation where the four node, and I'm not mentioning SMC5 and 6 here, which also has a role in this. That's well known. But these four different SMC complexes are all active on the chromosome. And they move around with an extrusion speed of about 1 to 3 kilobase per second. That's extremely fast. So it is inevitable that these machines will run into each other. Now the question is, what happens if they do? I must say, back in 2018 when we found this structure for the mitochondrial, we weren't completely comprehensive because this is only one sister chromosome, but we know that there are two sister chromosomes. And we know that in between the sister chromosomes there is cohesive that was installed there during S phase to keep the sister chromosomes together. So this is a more accurate picture of the situation in the entry of mitosis. We should probably replace the picture on the right by this picture where there is now cohesive, condensed one, and condensing two active on this chromosome. During this period, extrusive cohesion is known for working interface has been produced. So that is the role of these four components. And to go into it more deeply, I'm not going to explain the whole system that Congreso has developed, which is amazing. Basically, this chemical genetics that Bill talked about as well allows us to synchronize cells at the onset of mitosis. And then, by washing out the compound with liquid clot, we can walk into mitosis synchronously, which is for these bulk assays like IC. Very moist, and we have a very nice purity where you can see what's happening with the folding of the genome. This synchronous entry into mitosis is fifth. The other thing that is pivotal for this project is that we can use the oxygen diagrams that were developed by Congreso lab. And this is the cheap. Congreso is the diagram machine. I am struggling to do the high speed for all the cell lines. We have something like 70 to 100 different samples still in our freezer of different cells generated by Congreso. I never had the time to have that, and I was wanting to talk about money. So what Congreso did is he generated in his own diagram for SNC2, which is grades both of them part of the complex, but the complex also comes off the chromosome. So first, the SMC2 diagram results. I think this is something that's also pointed out by Phil. In wild type, you see that after 30 minutes, this is the time point to the right, you see mitotic, passive mitotic chromosome structure. If we add the auxin and basically degrade SMC2, the cells don't get, they do condense. So there are no condensers. What's in the name? There are no condensers. The cells condense. But from our IC, we can make compared to the wild type, where the structure gets lost during mitosis. So this is basically going from G2 into mitosis, and what Ed talked about previously is basically going from there on back into G1. So we're looking at entry into mitosis. In time, these little video snapshots of 2.5 minutes. That's remarkable. You have to really have an experienced cell biologist to be able to do it. That 2.5 minutes is the washout of the compound, the centrifuge, and everything until you fix the cells and you can analyze it. So this is very quick work with a lot of pockets of cells, and I wouldn't want to be anywhere near the whirlwind that is cool probably when she's doing this study. But I'm very happy to have all this happen. So, as said, the structure seems to disappear in wild type and not in the SMC2 AID. And this is recapitulated by looking specifically at groups, where you see in the wild type on the left, the structure disappears, the group disappears, and at the top right, with the oxygen added, the groups remain. We can quantify that. Percentage-wise, you can see that the SMC2 AID has a less small structure for compartments than does wild type. And these dots that we're talking about here are generated by cohesion. So the next question would be, if the dots remain in the absence of condensate and they get lost in the presence of condensate, can we model the interplay between condensate and cohesion? They're bumping into it. This is brilliant work by Samir Abraham and Ian Bernie's lab. We can model what would happen before the same situation. What happens if the condensate bumps into the cohesion? Does it unload? Does it push the cohesion away? Does it bypass the cohesion? Or does it stall? The four distinct options that we explored by modeling. And these are the maps that came out of this model. And what you can see from this, and I will show you in the next slide, is that if you unload or push the cohesion, you recapitulate the maps that we saw in our high school. If you bypass or stall the cohesion, that's not the case. So bypassing and stalling are happening, which basically means that extrusive cohesion is unloaded by condensate. And so this is our first rule of engagement. When condensate encounters an extrusive cohesion, the extrusive cohesion is removed. Then we thought about cohesive cohesion, so the cohesion that is in between the sister arms. This is another great picture in microscopy by Kumiko, where you can see in the dotted yellow box that the red signal coming from SMC2 is the axis of each sister, and in between you see this green signal, which is cohesive cohesion. And if you quantify this by taking a path through both axes, you see that there are two bumps for SMC2 as being the axis of the sisters, and in between you clearly see that that's where the cohesion is located. Here comes Samir again. So now that we know that this is what we should see as evidenced by microscopy, we can try to model what happens if condensate bumps into a cohesive. There are basically two options, because we know that the cohesive region does not get evicted. We know there are basically two options. It either passes the cohesive, leaving the cohesive region in the loop, or it stops with the cohesive. Cannot do a thing. This is what the model predicts the chromosome would look like, and this is what the plots would look like that go with it. As you can clearly see, it is the bypass cohesion that fits the microscopy found by Congreso, which basically gives us our second rule of engagement, that when condensing encounters a cohesive cohesion, it'll step over the cohesive cohesion. Cohesive cohesion ends up in the loop, so not on the axis, in the loop. And so from there on, I mentioned also by David, we have now an option to degrade specifically one type of condensing, both types of condensing, or cohesive. So we try to figure out what happens in combinations of removal of these compounds. So the first situation, if you take out SNC3, cohesive, you have condensing 1 and 2 left. Then there's a situation with condensing 2 only, condensing 1 only, or no extrusion at all. And in the contact map, you see that results in very different types of distance decays, but I don't have to show you this distance decay for you to see from the high sigma themselves that the results are clearly different, that these motors, removal of these motors results in very different heat. And most strikingly is this condensing 2 only situation, where you have not a single, a second, but also a third and a fourth diagonal, which indicates that over a certain distance, the length of these loops allow them to move in a vertical way along the axis, and touch upon loops that are basically 1 step or 2 step of the helix below or above. After Samir did his modeling, we would be lucky to have Anton Laboratorio as part of our team, because he is very good at polymer. And this allowed us to dig in deeper on these situations where we have only 1, 2 condensers or cohesive, to see what the effect of having only a certain subset of the motor around, what the effect would be on the shape of a microscope. So what Anton does is he takes the distance decay maps, tries to come up with polymer models that fit the distance diffraction for these maps. And then he creates high C maps that very much look, where the model looks very much like this. And what we learned from, and this is a tough word in our paper, we can read more about it, but the most important thing that we learned from this type of model is that the helicity that we, in our 2018 paper pointed out and found, is not at all regular. It's not like a staircase. There are differences in the shape. There may be buckling, there may be switching of helicity, but it is not regular at all. Another interesting thing is that we found is that there is a small spacing between the condensing and 1 and 2 groups, and that the cohesion between the sister, is what constrains the chemical pattern. One important thing though is that both condensing 1 and 2 groups are always consecutive, and that condensing stop when they run into each other and they do not pass or unload, giving us our third rule of engagement. And I will summarize it now. The first one is that when condensing encounters an extrusive cohesion, the extrusive cohesion is removed. The second thing we found is that when condensing encounters a cohesive cohesion, condensing steps over this cohesion. And then having the cohesion end up inside it, creating what we saw is like a less perfect image. And when condensers encounter each other, they stop and they both stay. And I'd like to finish up and again emphasize that this has been a remarkable collaboration that has kept me employed for 50 years now. The labs of Bill Decker, has his own lab now, and he did taken a long time, but I think we found some remarkable things. Not to mention, I think, maybe more than a scientific friendship, we have a very nice work together. A very happy time in my week on Wednesday mornings to call Edinburgh and Vienna every week. But those were the good things. Of course, most of the lab work has been done by people in the labs, and I would especially like to mention Congreso, amazing amount of work, and Celular, and Fernanda as well, whose data is specifically not sure because she's been on after, but very much of it.
Thank you very much. Yes, just a second. So the microphone is coming.
Thank you. So what I'm wondering is, how does it know when it's exclusive versus cohesive? And is there a molecular difference in the two complexes, or is it the cis versus trans conformation?
Right. So as you saw, I tried to depict them. I mentioned them differently, but I tried to color them the same. The components of the complex, as we know, as far as we know, are the same. But there may be assimilations on SMC1. There may be different different changes in these complexes that allow the condenser to pass when they kick off. With that, maybe give it the time to get it. So there are some different things at play that are more detailed that we are currently thinking about and researching, but it's not at all clear how that can be.
Well, that was really interesting topic. Really enjoyed it. I'm very curious if you have like a condensing 1, 2 all together or none, right? Like there's a high C of the 4 conditions, 1 alone, 2, and 2 alone, and then none at all, right? And what happens, what will look like in the absence of cohesion on top of it?
I'm sorry, so you have to, I'm trying to stand in front of the.
Yes, you have a slide that shows the high C map of condensing 1 alone, 2 alone, and on the mitotic console, and then both, and then none, right? So you see beautiful pattern differences. And what if on top of that you remove cohesion?
So one of the experiments that we did, I say we did, but mostly Congreso, is specifically use deeply cohesive by doing the oxygen degradation in S phase and then let it come back to G2. And from that we could figure out that the helical path that was generated by the cohesive. The differences in the helical path are generated by the cohesive. I'm not sure that completely answers your question, but yeah, sure. Love to talk about that.
So one of the things that was really made Anton Goloborodko, so Anton Goloborodko is this guy who sits there in the Zoom call with Python open on his screen. He's programming away while he's talking and showing things up, throwing things up in real time. But one of the things that he was really pleased about is he came up with a very nice polymer model for chromosomes with only condensing 1, and a very nice polymer model for chromosomes with only condensing 2, and there were differences between these two. And then you add those two models together, and you get the actual model you make when you look at the structure when you have them both there. Right? So they really can be separated. Thank you very much, Johan. Okay, so while Fernanda is getting ready, I met Fernanda